let me start by thanking the organizers, Martin, Weng, and everybody else for inviting me here to a series of very exciting and stimulating workshops. So I know from yesterday afternoon's discussion, maybe I will excite lots of debate about enhancers and their target genes. So I decided that might be a better topic to talk about. So let me start by saying that we have been working in the ENCODE Consortium for quite a number of years. And all this work has been done in the context of ENCODE. And as part of the ENCODE, there are the goal of the ENCODE is to catalog all functional elements in the human and mouse genome, and to develop freely available resources for the research community. And I borrowed this slide from NHI, Mike Payson. And basically here is how the consortium looks like in terms of organization. So there are data-producing groups who produce a large variety of data: RNA-seq, ChIP-seq of TFs and histone modifications, DNA methylation, DNA hypersensitivity, RNA binding proteins. And then there is a data coordination center that takes all these data and also perhaps equally importantly, the metadata associated with these data sets, put it in a unified place so that people can easily query the data and the metadata. My role is I'm heading a data analysis center that performs a lot of the analyses that are required by the consortium to be useful. And one major charge is to produce something called the Encyclopedia, which comes from I think E is Encyclopedia in ENCODE, which is a collection of analysis results that might be useful for the broader community. So people are asking, where is the encyclopedia? So far, ENCODE has generated thousands of experiments, as you probably saw from Bill's talk yesterday about picking the experiments. So hundreds of DNA-seq. These numbers are, I would say, actually more than these. So 200 plus is a little dated. So we have hundreds of DNA-seq and over 1,000 ChIP-seq for TFs and so on. And how do we integrate these very different types of experiments and assays to find functional annotations? So what we are working on to be included in this encyclopedia includes a number of things. For example, like expression of genes across a panel of cell types, transcription start sites, and all these enriched regions, commonly called peaks, from DNA-seq, ChIP-seq of histones and the TFs. And then chromatin contacts, for example, from high-C experiments, a lot of them conducted by Job Decker, and then CHIA-PET by Mike Snyder. And then also you have heard semi-automated genome annotations like SAGARs by Chrome HMM and Segway. And then today I'm going to talk about these two components. One is candidate regulatory elements, especially enhancers, and also the target genes of these enhancers. So the goal of predicting these enhancers include a number of ways, because enhancer prediction has been pursued very actively by quite a few groups so far. And our goal for predicting enhancers as part of the encyclopedia has a number of distinct goals. For example, the first one is we are more in favor of unsupervised method, so that we can apply the method broadly to a large number of cell types in both human and mouse. And then the second goal we like is to try to incorporate multiple epigenetic features, such as DNase-seq and K27AC, which have been shown to be really good markers for enhancer activity. So we want to apply broadly to cell types. So we actually have been working on this. My lab has been working on this for quite a bit of time until we realized that we really should work on these methods using ENCODE mouse data. So the groups in ENCODE who work on mouse have come up with a really nice collection of data. So very nicely formulated matrix of developmental time points and cell types in mouse with a large panel of experiments. So histone marks, ChIP-seq have been made by Bing Ren with a panel of histone marks, and RNA-seq data have been produced by Barbara Wald's lab, DNA methylation by Joe Ecker, and then DNase-seq, not for the whole matrix, but for a subset of the cell types and time points by John Stam. So here is the matrix I'm talking about. So there are developmental time points, embryo 11.5 days, and so on and so forth. P0 is the day of birth. And with these first three categories of data, the whole matrix will be populated by the end of this stage of the ENCODE project. You see some white cells here, but those are still being filled in. DNase will not be performed on this whole matrix, unfortunately, but it will give us actually DNase is more available for human data. So we really wanted to test as many features as possible for predicting enhancers. So we focused our effort on 11.5 day data sets. So once we have formulated our method, we can then apply it to the entire matrix and then predict enhancers in a cell type specific and time point specific manner. So in addition to these data, we also have another group participating in ENCODE, Land Panacchio and Axel Wiesel. They have been building this VISTA database, which has a lot of experimental validations of enhancers using transgenic mouse experiments. And I think you have heard about this kind of data from Nadav the day before yesterday. So, so you probably are familiar with that now. So VISTA has over 2,000 regions that have been tested. So what they do is they clone the region and micro-inject into a mouse egg and then put it into a pseudo-pregnant mother, and then at embryonic stage, whatever the time point you want to assay, very commonly 11.5, and then you will stain for the reporter gene and then to see whether or not the region that you have designed is functional at that time point. So this is laborious experiments, but one thing that's high throughput about this experiment is you can assay the entire embryo. So if this construct is active at any tissue type in the embryo, you will see it. So it's parallel in terms of cell type. So so these 2,000 regions are active in selected types of cells, and for limb and 4 subbrain regions and heart and neural tube and whatever, there are hundreds of active enhancers. So we decided to take these data set, albeit may have false negative regions, but it's highly accurate for the regions it has tested. And we decided to use this as our training and testing data set for developing enhancers.
So Zhiping,
yes?
Only 1 in 10 of the regions they test shows enhancer activity. How are they choosing the regions?
No, I didn't mean that. So for each of these cell types, we have hundreds of regions that are active.
Oh, I see.
So collectively, they have tested 2,000 of them. So we just take the ones that are active, say in limb, treat them as positives, and then the rest of the regions are negatives, even if some of them may be active in heart, for example. Okay. So here is a gallery, and you can see that these cell type specificity is pretty distinct. Yes?
How many of the enhancer tested negative in all the tissues?
Oh, so in their data set, because they're testing regions that are likely to be active, so they don't have a lot of them that are negative. But if you just randomly take regions in the genome and test them,
that was sort of identified from the whatever experiment, and then they chose to test.
Correct.
So what proportion is actually totally negative?
I would say that in there, maybe 10 to 20% are completely negative. But these, again, these are regions that they had reasons to believe that might be active. They haven't tested a lot of the regions that they thought might be negative to begin with.
Yeah, no, only those are the high confidence ones. But even among high confidence.
Yes. Still, some of them are completely negative. Yes.
Well, and even those could be active at different times before the experiment is done or after.
Correct. Could be active in other time points of development. Yes. So taking all the caveats in mind, so this is still a highly valuable data set. So for testing the algorithm, we were actually, so maybe I should spend a half a minute talking about, like, at the beginning we were very ambitious. We actually organized like a blind test for all the ENCODE members to participate, and everybody submitted their predictions. And people were using all kinds of features, thousands of input features and doing machine learning on all that. And then after the evaluation, we realized that features that are specific to that cell type and that time point are by far much more predictive than other features that are produced for other irrelevant cell types and time points. So we actually scaled back dramatically, try to build the method from more ground up and single feature approach. So I don't want to go through the whole process, but I just want to say that while developing a very simple and unsupervised method, we tested a number of things. For example, how do you center your predicted enhancers in the genome? If you can't do base pair resolution, you'll be overwhelmed by multiple testing. So we tested two choices. One is centered on the peaks of DNA's signal. I will give you some screenshots, and you will see what I mean by these peaks. They are actually quite peaky. And also the other choice is centered on K27AC peaks. And then how do you rank these regions, right? You can rank them by the p-value of a peak calling algorithm or by the raw signal of reads around the peaks. And so these are the two things we really spend time testing, and I think the conclusions are pretty broadly applicable. And as I said, they were multiple types of signals, and when I showed you the matrix and DNA methylation, other histone marks, and so we also considered them in a stepwise manner. So how do we test the algorithms? This is probably really trivial to you guys here, but basically we just used the VISTA positive and negative, and then the regions that we pick out, these peaks, we call them peaks, so either DNA's peaks or K27AC peaks, and then ranked differently using this confusion matrix to compute a precision recall, and then compare the methods.
Wait.
Yes?
The negatives are then far fewer than the positives, whereas in real life the negatives are far more abundant than the positives, right? So the precision recall curve is going to be wrong?
So usually for each cell type, there are about 300 positives and then 2,000 minus 300 negatives.
I see, because per cell type.
Per cell type. This is specific to each cell type. I mean, it's skewed, but still not as skewed as in real life.
Not a realistic skew.
Correct. So that's why we're doing PR, precision recall. Right.
So this is not really predicting the enhancer, but it's predicting in what tissue the enhancer is active. right? I mean, because you're choosing from things that you sort of already know are enhanced. 2,000 they are all enhancers. Only some are not active in that tissue. That's what you want to predict, right?
So not all 2,000 are active in one of the tissues tested. Some of them are negative everywhere.
Okay. But
so I agree with your saying, it's more a tissue specificity prediction. I mean, I don't know if it's more or less, but we're trying to predict both tissue specificity and whether or not it is an enhancer in one shot.
Yes.
Right. Okay. So, I mean, if you have confusion, you can ask me later again. So here is a typical PR curve and precision recall. And so here you can see that we have different ranking scheme and then different ways of focusing on which kind of signal. So the reason that I'm only showing DNAs and K27AC in this plot is they are actually the best single features. We have tested exhaustively each of the features beforehand, and then these two come out to be the best single features, and then we're trying to combine them. So as you can see here, there is a straight line here. So those are the regions out of the 2,000 that are negatives in that particular tissue type, hindbrain here. So we have done this, and then the results is after all the testing, we came up with a very simple non-supervised approach. So if we center our predictions on DNA's peaks, and it works better than on K27AC peaks, which kind of makes sense. Do I have a figure? So next figure probably shows. So hang on with that thought. And then the other one is we have incorporated additional data such as DNA methylation, K4ME123, K9AC. So these are all active marks. And then these two, K27AC, ME3, K9ME3, these two are repressive marks. If you include these using like simple approaches such as linear regression, logistic regression, they did not improve the performance at all. I'm not saying that they're not helpful, but just in our evaluation scheme, it didn't help. So here is the method is actually extremely simple. You have DNA's peaks. So for example, you can see in this raw signal, they are very peaky. So you can pick the center of the region, and then you have a K27AC signal. So from here, you can see that why picking the peak of DNA's signal is more effective than K27AC, because K27AC is on histone. It's a histone mark, so it's on histone regions. And usually these enhancer, TFs, they bind at open chromatin regions. So DNA's peaks are more accurate. They're actually at the troughs of K27AC signal. So we take them, and then we individually rank them with their signals, and then we just simply average their ranks. And then at the end, we predict an enhancer region that has centered on the DNA's peak, but has the sides of the K27AC peak. So at the end, here is our prediction. Okay. So here is an example, predicted region that is beautifully at the DNA's peak, flanked by K27AC peaks. And then this region happens to be one of the positives in neural tube. So you can see if you, this is the region that was tested in VISTA, overlaps almost perfectly, but it's not the same region. So if you take this region and then inject it into mouse, you see a strong staining in neural tube. So I have, I don't know how I'm doing with time, but there is a visualization tool that you can try it out with human and mouse. So for example, you can pick each cell type with data available. For example, any of these, you can check one of them. And then, for example, for time, I just picked one, like the 10th prediction. So this is how it looks like for each one of them. So very often you will see that there is a DNA's peak basically they have this peak and trough relationship with K27AC. So here is our prediction, and the overlay doesn't work quite well because the DNA's signal is on a very different scale as the K27AC. We're still tweaking that, maybe using the oxygen transform, what you guys were talking about, Alex was talking about yesterday, and other transform on the DNA's data, bring them more to the same scale so you can see their relationship.
Roughly how many peaks would you then typically predict in a cell type, genome-wide?
That's a really good question. We don't have a good handle on that yet. We truncate them at 20K. And usually you have like 100K DNase peaks and roughly 30 to 50K K27AC peaks. So that we're talking about tens of thousands of regions in the genome. Oh, okay. Quick then. So we applied it to human data sets, and then as you can see, in human we have worked on ChromoHMM. You can see a lot of the regions we predict are strong enhancers. And also they are bound by P300 in GM. So these are all cell type specific. And if you take the top regions we predict, applied it to roadmap epigenomics data, and you can see that they cluster cell types very well. So these bands of cell types are, you can see they are both, even adult and fetal cell types, they cluster pretty well, just as a sanity check. And also, a lot of people will be interested in knowing, like GWAS SNPs, do they land in these enhancer regions? So this is one particular example that's associated with Crohn's disease and inflammatory bowel syndrome. And you can see that this overlaps, if you look at the whole panel of cell types that we have in ENCODE, the first one that comes up is a 73rd ranked peak in primary T cells. And if you look at this peak, and it has a very strong signal, so this SNP is exactly at this DNase peak, ranked by K27AC signal. And so one natural question is, where is the target gene of this enhancer? And more generally speaking, where are the target genes of our predicted enhancers? So in this case, people have some idea. People think it is this PTGER4 gene, which is 150 kbs away, okay? And moreover, there is a high C link between the enhancer and that promoter. Well, you may think this is the closest gene, but actually if you look at the annotation, there are a lot of non-coding genes in between. So it's not very obvious that this would be the gene you should pick. So in general, we want to predict target genes of regulatory elements like our enhancers, right? So one approach that has been used a lot in the field, one very simple approach, is basically to correlate the signal between the gene and the enhancer across the cell types. So the idea is if these two regions are on in the same set of cell types and then off in the remaining cell types in a way that they are correlated across cell types, perhaps that enhancer regulates that promoter. It has been commonly used. So there are a lot of things you can correlate because we are very rich with different data types. We have DNase you can correlate. You have K27AC you can correlate. You have all kinds of things you can correlate in ENCODE, right? So we set out to do very simple things. We just say, okay, what's the best thing to correlate, right? How well do you do if you correlate them? And obviously, there are other groups in ENCODE who are using much more complex data integration and machine learning methods, which I'm not covering here, but I just show you some really simple results. So in order to evaluate how well you can do, we resorted to high C data, given all the caveats of it. And then we tested using this particular high-C data set because it's a promoter capture. So that normally if you see high-C, you see very sparse. The loops are very sparse. But if you use a chip to capture out all the links that have promoter involved, that involve a promoter, you see there's a big enrichment of things that come out of the promoter.
Protein was targeted in the chip, in this case?
So you designed oligos to capture promoters, and then sequence only those links that you pull out. Okay, so we took that data set and then calculated the distance between promoter and a non-baited fragment, possibly enhancers. And you can see majority of them are under 1 megabases. And then, so we established a training, testing, and validation data sets using data from that paper. So if you have these enhancers that we predict in that cell type, we take all the links within 1 megabase if the other link is a single promoter. I'm skipping this one in terms of point, interest of time. And then we correlate all kinds of signal between enhancer and the promoter. And so how well did we do? Not very well, unfortunately. So you might be depressed over a ROC curve of nearly 0.5. You can use DNase signal to correlate, and you can use K27AC to correlate, or you can use our approach of averaging the ranks to correlate. You don't do very well. The problem is because there are so many regions in the genome that you predict as enhancers. So what do we do? Okay, this is the PR curve, corresponding PR curve. Also doesn't look very encouraging. Okay, so what do we do? So obviously correlation by itself is people use it a lot. So this is a wake-up call. It by itself doesn't do very well. So what else can you add? So you can add the distance between enhancers and target genes. You can add the expression of the target gene, right? If the target gene is not even expressed in your cell type, why would you predict it? That seems not sensible. So you can add all these features. So I'm happy to report that if you add these features, just simply do a random forest, now you actually do much better in terms of prediction. And this is very preliminary results, and you also do better in PR curve. So this is actually a very tough problem to predict because roughly less than 5% of the links are true positives, and more than 95% of them are actually negatives. So you can also look at the feature importance, and the distance between enhancer and promoter is actually the most important feature. And you can see that these features indicate whether or not the enhancer itself is active in that cell type, whether or not the promoter is active in that cell type. Only later correlation contributes. So, so for these, let me just wrap up. So for this direction, we hope to incorporate additional training and testing data for predicting enhancers because obviously the VISTA data set is very small in size, and that might be the reason that we cannot incorporate more sophisticated models. We test these features, and then also predicting target genes remains a major challenge, and what additional features can we incorporate into the machinery, and then we're also defining other types of regulatory elements. So can I just have 1 minute to showcase another method? I promise to be fast. So it just occurred to me that from the talks yesterday, so I should probably show you that we have this tool called FactorBook. I think Eric mentioned about ChIP-seq of ENCODE data. So this is a method that has all the released ENCODE ChIP-seq data. It's in a matrix, and so for each one of them, you can have human and the mouse. So these are the experiments that have been released by ENCODE. So for each one of them, and you can sorry, that's not. So FactorBook. So you can look at one of the TFs you might be interested in. So maybe Trevor, you were talking about what was IRF3?
Sure.
So, so there is so maybe the TF motif enrichment might be something interesting for you. So these are the results of mean chip. So top 5 motifs that were identified by mean chip, and we filter them. But obviously, motif detection is quite a tricky business. So we have implemented it this way so that people can vote for the motifs. If you like it, you can give it a thumbs up. So this looks like a real one, even though it's showing up in 200 out of the top 500 peaks, and not as prevalent as the top one. So maybe I'll give this one a thumbs up. But is this a real motif? It looks good to me. I don't know. So I'm hoping people who use this resource, who know the TFs, will give us some feedback. We can also let people put comments. That's in the works.
So that's NFY.
Yeah, this one.
It's the cat box.
You see, that's useful. So put your comments when you look at that. And why it should be here, whatever you know. So we'll allow people to put comments. So all the released ENCODE ChIP-seq data sets are included in here, and it's automatically updated. It's linked directly to the metadata, and there are all these other features. So you can look at them across the genome, and you can see whether or not other TFs bind in a correlated manner across the genome. And then you can go directly to the data set in ENCODE to see what it is, where we got it from. So anyways, I hope this will be useful for people studying TFs. That's it. Thank you.
One quick question. So I didn't understand how you're using the promoter capture high-C to do the validation. So how many promoters are you targeting? And presumably you can't target all the promoters, or do you target all known promoters?
So in that experimental paper, they targeted all the promoters. They designed this array to capture the promoters, all the promoters, genome-wide.
Okay, and so then when you presumably find contacts that involve a promoter and something else.
Correct.
There's got to be some kind of statistical confidence estimation for those things. Otherwise, you're going to have a bunch of spurious stuff. So how do you say what you think is a real enhancer based on this? And I realize it's going to be noisy, enhancers in quotes, but you still need some kind of confidence estimation before you, anything that, there's lots of spurious high-C contacts.
So from their data, we get all the links from a promoter to any other regions in the genome. And then we have predicted enhancers.
You get all the links regardless, even if it's only seen once in a single high-C read pair, you call that
That is a good point. I have to ask Jo exactly what kind of cutoff she applied. But some sensible cutoff.
It's got to be a distance-dependent cutoff also, right? Because of that genomic distance effect.
The cutoff, we didn't apply a distance cutoff. We probably applied a number of reads cutoff.
I'm just saying the number of reads cutoff should depend on the distance between the sites.
Somehow the people that did the capture had some way of doing it, right?
Yes, we just take we didn't manipulate their data.
Some statistical tests for this kind of stuff.
Right, but as you can see from the histogram, it does taper off very quickly as a function of distance. What?
More of a comment. I think one of the origin of the difficulty of designing a classifier that also uses DNase accessibility is that where the chromatin will be really accessible to DNase, you expect very few nucleosomes. So the histone markers, we're not going to see them where we have the DNase peak.
That's right.
I mean, I imagine that the nucleosomes have been rolled outwards. In between, it's DNase accessible.
Correct.
And the histone marks are on the flanks of this region.
Exactly. We're trying to take advantage of these kind of. That's exactly right. Yeah, that's why we combined the DNase with the K27AC, and we show consistently across all the cell types we tested in mouse, there is an improvement.
Did you look at all at conservation of the region to
No.
incorporate?
We can. We tried early on, the help from conservation is very small.
Very small.
Very small. Helps a little bit.
Okay.
Okay, let's thank the Zhiping again.
Thank you.